Okay, now we are discussing about bacteria last time, of course, and this some portion I think covered, is not it? See, one thing we have learned, the bacteria we know it is not visible to the human naked eyes, but at least one bacterium or two bacteria that has been discovered, and now it is visible to the human naked eye. This is some exceptional cases. Otherwise, bacteria are not visible to the human naked eye. Thiomargarita namibiensis. This is the bacterium that is observed in the bottom part of the sea. Now it is a very big size, 750 micrometer, something like that. It is quite visible to the human naked eye. These are some exceptions. Is it otherwise? Bacteria are not visible to the human naked eye. Okay, coming back to the basic question. What is bacterium? Prokaryotic organism. Unicellular. Many things you can say. If it is asked write about bacteria, then you can write so many things. But what is bacterium? Bacterium is prokaryotic unicellular microorganism. Because if you mention prokaryote, it will tell so many things. Because you know the characters of prokaryotes. Isn't it? It depends on what amount of things required actually to answer the question. What is bacterium? Bacterium is a prokaryotic unicellular microorganism. Okay. Then if you mention prokaryote having so many characters, can you mention some characters of prokaryotes? I think you have studied in your higher secondary program. You answered this question also.
Primitive nucleus.
Primitive nucleus. Good.
Organelles are not membrane-bound.
Membraneless organelles. It has then 70S ribosome. Very good then. Like chromosome number, how many numbers of chromosome present in case of prokaryotic organism? You have studied in your higher secondary program, isn't it? You have answered this question also. You have passed out this higher secondary program, answering this question. And you did well in the biology, I believe so. Prokaryote having so many characters. Okay, if you list out, it will get around say 20, 25 characters are there. Prokaryotes and eukaryotes. We have very well marked, well difference between these two, prokaryote and eukaryotes. Variations are there among prokaryotes also, among eukaryotes also. But basically, what they differ, what way they differ, we know the well established characters available. So if you mention bacteria prokaryotes, you can answer so many things associated with prokaryotic character. And these are representative of the prokaryotes, representative of the bacteria, or any other prokaryotes that you are mentioning. Variations are there. Say, although prokaryotes having cell wall. Somebody in your answer script, somebody you have written prokaryotes lacking cell wall. This is not a true statement. Majority cases, prokaryotes having cell wall. But we have the example of prokaryotes lacking cell wall. Cell wall less prokaryotes. Example is, can you name the bacterium which lacks cell wall? Wall-less bacterium. Wall-less bacterium. Have you heard about mycoplasma? Have you heard about phytoplasma? Spiroplasma? These are wall-less bacteria. We are going to discuss about this in next class, not now. But this is the example, that bacterium lacking cell wall. So in this way you will get variation among the prokaryotes. Some characters vary, accordingly it is named different way. Suppose wall-less bacterium, we know this is mycoplasma. But in mycoplasma you will get variation. Some are spiral form, some are round spherical type, some are filamentous form. Accordingly different names given. Spiroplasma, this is one type of mycoplasma, but it is spiral in nature. Phytoplasma, again phytoplasma, this is mycoplasma causing disease in plant. Phyton means plant. That is what is known as phytoplasma. Mycoplasma actually animal mycoplasma, which is causing disease in animal cell. So accordingly we are naming it that the different prokaryotes we have. Anyway, now this the bacterium is a prokaryotic organism that we know. I think we have discussed about it. The size of bacteria, this is important. See, size of bacterium, as you know, this is given, the smallest size is 0.2 micrometer. 0.2 micrometer. One question asked in your midterm question paper. What is the resolving power of the microscope? light compound microscope that we have. 0.2 micrometer. 0.2 micron or 0.2 micrometer, this is the resolving power of the microscope. You can correlate this resolving power with size of the bacterium. Smallest size of the bacterium on an average, the smallest size of the bacterium, what is the smallest size? 0.2 micrometer. You can correlate these two things and you can remember it a better way. How can you remember the resolving power of the microscope? You can remember the smallest size of the bacterium, it will give the answer of the resolving power of the microscope. Either way, if anybody asks what is the smallest size of the bacterium, if you do not know about that, but you know the resolving power of the microscope, you correlate with this information with this resolving power of the microscope, vice versa. The smallest size of the bacterium is 0.2 micrometer, resolving power of the microscope is also 0.2 micrometer. What relation they have? Then you can define resolving power of the microscope. Why it is resolving power of the microscope, 0.2 micrometer? Then you have to know what is resolving power of the microscope. That is asked in the question paper. I have asked this question paper deliberately because so that you, what concept you have developed, what is resolving power of the microscope? Forgetting the other points. It's very ordinary definition because we are ordinary people. We have to remember the things in a very ordinary way. What is the ordinary definition of resolving power? You. Ordinary definition of resolving power of microscope. No, that is there, but this is not the ordinary definition as such. Very ordinary definition of resolving power. Sit down. Smallest?
Size of organism visible.
Visible, good. Now, that is the answer actually. Smallest size of the organism visible. The smallest structure. Smallest structure or the microorganism or organism visible under that microscope. Forgetting other definition. No need to remember. Okay, ordinary way let us remember this definition. Smallest structure visible. Why smallest structure visible? That is not our concern now. But definition, as per definition, the resolving power indicates the smallest structure visible under that microscope. Why and how? This is not our concern now. We have to remember smallest structure visible. Now, what is the smallest structure that is visible under light compound microscope? 0.2 micrometer. If it is below 0.2 micrometer, it will not be visible. Why it will not be visible is another chapter. But try to remember, try to develop the concept that resolving power will mean ordinarily the smallest structure visible. And a smallest structure that has been found out as 0.2 micrometer. And this is also the smallest size of the bacteria. On an average, smallest size of the bacteria is 0.2 micrometer. See, by knowing the size of the smallest size of the bacteria, that is 0.2 micrometer, also we know the resolving power of the light compound microscope. Because as per definition, light compound microscope, the resolving power of the light compound microscope means smallest structure visible. In this way, you correlate some facts so that you can remember. If you have the correlation with a different facts or different terms or different words, that we can remember in a better way. Always try to make correlation so that you can remember. Separately, you need not to remember. Because this definition that I have asked, of course, few we have written the smallest structure visible, or wavelength divided by my numerical aperture, or the power of the lens system to dissociate very closely associated point distinctly. Whatever you define, but ultimately, practically, it is the smallest size that is visible under that microscope, 0.2 micrometer. Say, we have the microscope, ultraviolet microscope. What is resolving power of the ultraviolet microscope? 0.08 micrometer. 0.08 micrometer means here we can see the smallest structure visible under the microscope, 0.08 micrometer means it is below the size of the bacteria. So with ultraviolet microscope, we can see the smaller than the bacteria, because it is having resolving power 0.08 micrometer. In case of, say, electron microscope, 0.0001 micrometer. So in this way, see, smaller the value, greater the resolution, greater the resolving power of the microscope. So this is that we have to remember. I think you should not forget in your life. See, how do you forget? If you develop the concept and making the correlation, I think you never forget this thing. If anybody asks tomorrow, what is resolving part of the light compound microscope? Because the common microscope we are using in our laboratory, all the laboratory of the country of the or the world, because this is the very available microscope we have in our lab system. So always it is having 0.2 micrometer resolving power, and this is the smallest size of the organism, that is bacterium, because we can see bacterium with this microscope that we are handling. Okay, size of the organism is important. And now parts of this, the bacterium, say one bacterium that I have shown you last time, I think one, this diagrammatic represents of the bacterial cell. The cell, it parts of the cell envelope, it has one envelope cover, cell wall. The following parts of the cell envelope are usually recognized. What it is the surface appendages, flagella, pili or pilus. Pilus is the singular term. And filamentous appendages, very, very rare, it may have. And this is surface appendages. You know appendages? Appendage means additional structure which is not required for life process completely. Without this, life can survive. They are known as appendage. But this bacteria also having some appendix and some structure like flagella, pilus and filamentous appendages. Also it has surface layer, slime layer and cell wall. See, cell wall is there. As you know, the bacteria having cell wall. Above the cell wall, surrounding the cell wall, it has some envelope which is known as some structures we have to discuss about the next. See, flagella. Bacteria having flagella. Bacteria can move like animal. Because of bacteria like animal. The Leeuwenhoek, 1676, when he found like animal bacteria in film of water or mud or anything, rotten material he observed with his small microscope which he constructed. By seeing the movement of this bacteria, he thought that those were animal. Then he coined the term as animalcules. As you know about that, animalcules means animal-like. Because of this reason, bacteria can move like animal. The animal can move, movement is there. But bacteria can move with the help of flagella, that we know about that. The most motile bacteria possess long up to 20 micrometer or then 20 nanometer the thickness is there. A helical appendage called flagella. Flagella is there. And flagellum has no definite membrane. Okay. Arrangement and number of flagella. You see what way flagella attached to the bacterial cell, or numbers of flagella, placement of flagella. See, flagellation. Flagellation is one of the important diagnostic character. Presence or absence of flagella. It is not like that all bacteria having flagella. Then you can ask me the question, how that bacteria can move? Even without flagella, bacteria can showing its movement to some extent. Gliding movement, as for example, I cite one example of gliding, you know. But it needs some external force. It cannot move its own. It is not like so. Inert matter cannot move itself. When you exert extra pressure or external pressure, it can showing its movement. But some external force required. Bacteria can generate external force. Later on you will learn higher courses how it can generate such types of force in their system by which it can glide. Gliding movement is there. A flagellate one. It has no flagella, but it can showing its movement. Sometimes it can jump like a frog. Because bacteria also can jump that way. Without flagella, bacteria can move this way. So some movement is there without having flagella also. But otherwise it can move with the help of flagella. And flagella entirely consists of a protein called flagellin. You have to remember this. There is some important points here we mentioned from this lecture. Okay, it has a protein that is known as flagellin. See now how it is named. This is important. You can expect this question in examination. See, when flagella absent. See, flagella may present normally bacilli from bacteria and spiral from bacteria, very common. And coccus sepet bacteria normally does not have flagella. But see, when flagella absent, we call this bacterium as atrichous bacterium. Atrichous. Trichon means flagella. A means not. They do not have any flagella. Monotrichous means single flagella. You know, if you consider it is a rod sepet bacterium, the diagram is not good actually. See, in a rod sepet bacterium, it has flagellum in one end. It is known as mono means single. It has single flagellum. And lophotrichous means lopho means two. It has two flagellum in two ends. It is known as lophotrichous. Okay, and then a cephalotrichous. Cephalon means groups actually. Group of flagella in one end is known as having cephalotrichous. MP means both. MP means a group of flagella both ends. Normally spiral type organism having such types of flagellation. Group of flagella both ends. Group of flagella in one end, this is known as cephalotricha. Then flagella all along the cell wall, it is known as peritrichus. Peri means all the surface of the bacterial cell having flagellation. Now it will give certain diagnostic character. So peritrichous flagellation, if you find peritrichous flagellation, say one bacterial genus having this peritrichous flagellation is Erunia. Later on you will learn Erunia group of bacteria having peritrichous flagellation. Monotrichous Xanthomonas or Pseudomonas. In this way you will learn. Spirillum having amphitrichous flagellation. So by observing such types of flagellation, you can identify its systematic position or its identity. It helps to identify the bacterial group. And whether bacteria having flagella or not, that is also giving certain type of identity. Presence or absence of flagella and placement of flagella, where it is placed. In one end, both end, or numbers of flagella, whether one flagellum or more than one flagella. All these things taken into account while diagnosing particular bacteria. So arrangement of flagella like this way. Times we call it pili or pilas. Slight variations are there, pili and Fimbriae. Okay, these are some hair-like appendages like flagella, but these are not the flagella. Why these are not flagella? Because it has no role to play in movement. Then what function they have? See, Fimbriae present on cells of bacteria as filaments and half as thick as flagella. Is it thinner than the flagella? And shorter and much numerous. Numbers are very, very high as compared to the flagella. Help in attachment with other cells. The bacteria sometimes used to get attached with other structure or animal or anything. Assist pellicle formation, one of the reproductive mode, on the surface of liquid cultures and help in sexual reproduction. See, later on you will learn when you discuss about the sexual reproduction of fungi. Very, very rare occasion actually. Bacteria normally produce asexually, they through binary fission. But sometimes it exhibits sexual reproduction, and during that process, pilas help this. Pilas act as bridge between two bacteria, through which nuclear material getting transferred from one bacterium to the another bacterium. Because sexual reproduction, nuclear material must exchange. So that is possible with the help of, that is why it is written as sexual reproduction possible with the help of pilas or pili. It acts as bridge between two bacteria. See, filamentous appendages, those are not very, very rare. Such appendages are rare, thinner than the flagella and pili, and not known to have a specific function till date. Research is on what is the function of this appendage-like structure, but still not known. See, surface layer. Then next one. Surface layer. Now slime layer, let me explain. See, bacteria having cell wall, cell membrane and other organelles. Above this cell wall, surrounding this cell wall, it has one layer which is known as slime layer. Slime also known as capsule layer. What is slime? Slime is colloidal substance. It is an extra polysaccharide actually, polysaccharide in chemically. Now the cell wall is surrounded externally by a thin slime layer, which is colloidal in nature. It is a modified outer layer of cell wall. It is modified, means this slime layer developed due to secretion of certain material, metabolites secreted by the bacterium getting deposited in the cell wall, above the cell wall and from the slime layer. When it is very thick and conspicuous, it is called capsule. There is no distinction between capsule and slime layer as such basically. From slime layer, capsule layer modified. Actually, this is the modification of slime layer. Slime layer, in the very initial stage, it remains in a very semi-liquid condition. This is known as slime layer. And when it gets thickened and hard in nature, then it is called capsule. Now, capsule composed of complex polysaccharide. And it is about 10 micrometer or more broad, act as a non-vital and non-essential envelope around the cell wall. It is like non-essential part. Without this, bacteria can survive. But what more important function played by this slime layer, you will learn higher courses. Slime layer helps in pathogenic ability of the organism, of the bacteria. It helps in pathogenicity. Later on you will learn. Not now. Because it gives certain chemical signal to identify the susceptible host. And thereby they can recognize the sites and infection starts. It helps to recognize the host system. Later on you will learn chemical signal given by the slime layer. It helps that way in pathogenic ability of the organism. That is one important vital function performed by the slime layer. But when it turns into capsule layer, it provides physical defense. Because of this thickness, you cannot kill this bacteria so easily. What happened in case of tuberculosis? Oh, this is also asked in examination. Causal organism, Mycobacterium, it is TB of human being. Mycobacterium tuberculosis. Causal organism, Mycobacterium tuberculosis. Mycobacterium is the actinomyces actually. Now, what is this? It is a bacilliform bacteria, one type of. And this Mycobacterium tuberculosis having capsule, which is very, very thick. And because of the thickness, additional structure that develop surrounding the cell wall, the drugs, medicine or antibiotics, whatever you call, that you are using against this pathogen, against this bacteria, it finds difficult to penetrate this bacterial cell. Because to penetrate the bacterial cell, it has to penetrate first this capsule layer. And capsule layer is a slime, is a complex polysaccharide. And the drugs find it difficult to dissolve this layer so easily. It is not so possible. It is very difficult. So later on you will learn about that. So because of this reason, there are such types of bacteria very difficult to kill with the help of medicine. Likewise, there are certain bacteria, diphtheria toxins, Corynebacterium, diphtheria. Corynebacterium diphtheria producing diphtheria toxins. It is a very, very heat resistant bacteria. If you go on boiling for hours together, you cannot kill it. This is the situation. So it has defense mechanism because of this capsule or envelope layer. But this is coming to the cell wall. Now we have understood something about this envelope, appendages and all. Coming to the cell wall proper. Now what is this? It is a rigid protective structure and responsible for the shape of the cell. It is very clear. Where cell wall is there, it gives the specific shape of this cell, a specific shape of the tissue, a specific size of the organism. It is possible because of this cell wall. Now, it is not semi-permeable membrane, as you know about semi-permeable membrane present in case of cell membrane, but can act as a molecular sieve, preventing large molecule passing through. It can chip the large molecule. It does not allow the large molecule to go to the cell, although it is not the semi-permeable nature, but it acts as molecular shape. That is the one important vital function performed by the cell wall besides providing rigidity to the cell. Main components of the wall is a mucopeptide, you have to remember. Because what is the shape components of the bacterial cell wall? You may face this question, not in this course, any course, anywhere in future. It is a mucopeptide or peptidoglycan, whatever you call. Mucopeptide or peptidoglycan. This is the shape composition of the bacterial cell wall. Substance along with protein, to some extent protein, polysaccharide and lipid. Some amount of these biomolecules also attached with this mucopeptide. The thickness of cell wall ranges from 10 to 25 micron. This is the thickness. This much thickness is there in case of cell wall. See, cytoplasmic membrane. Beneath the cell wall, it has the cytoplasmic membrane. Now see, in case of cell wall less bacteria, see mycoplasma, for example, mycoplasma does not have cell wall. It has cell membrane. And there are certain bacteria, as for example gram-negative bacteria. Gram-negative bacteria do not have cell wall proper. What it has? It has unit layer membrane. 3 sheets of layers constitute one membrane, and this is the characteristic features of gram-negative. So it has triple unit layer membrane instead of cell wall in case of gram-negative bacteria. And there are certain bacteria which does not have cell wall at all, as for example mycoplasma. Now we can answer one question. See, you will find later on the mycoplasma insensitive to penicillin, but sensitive to tetracycline. Penicillin, that is antibiotic used to kill the bacteria which are having cell wall. Because the antibiotic is having a specific action. Sometimes it acts on the structural integrity, meaning structure, cell wall, cell membrane, something like that. Or sometimes it can act on the metabolic pathways, metabolism. tetracycline having the action on the metabolic pathways. Penicillin act on the structural integrity, means cell wall. It has specific action. It does not have any action on the metabolic action or metabolic activities. Since mycoplasma does not have cell wall, so there is no action of penicillin. Penicillin, with the use of penicillin, we cannot kill mycoplasma. Are you getting the point? So cell wall presence or absence will determine what type of antibiotics you are going to use against those organisms. Now, see, as a semi-permeable membrane controlling the flow, and it is a thin layer, 5 to 10 micrometer, lining the inner surface of the cell wall and separating it from the cytoplasm, because it embedded the cytoplasm, not the cell wall. Cell wall, the external barrier. And it acts as a semi-permeable membrane, as you know, controlling the flow and outflow of metabolites to and fro from the protoplasm. It is possible through the cytoplasmic membrane, as you know, in other cellular organisms. Electron microscopy shows the presence of 3 layers constituting a unit membrane. Because these, only in case of gram-negative bacteria, it has only this unit membrane. And chemically, the membrane consists of lipoprotein. That is what to remember. At least some biomolecule, what biomolecule present in each components. In case of cell membrane, it has lipoprotein, lipids and protein together, with small amount of carbohydrates. This is the shape composition of the membrane, cell membrane. Then cytoplasm proper. Now we are starting from the appendages area, then cell envelope, capsule, then cell wall, cell membrane, then coming to the cytoplasm. Now it is a colloidal system of a variety of organic and inorganic solutes in viscous water solution or soft gel. Always you know cytoplasm is a semi-liquid substance having this, and differs from eukaryotic cytoplasm, remember, in not exhibiting internal mobility. There is no protoplasmic streaming. You can differentiate between eukaryotes and prokaryotes this way also. And in the absence of endoplasmic reticulum or mitochondria. There is one question, say, bacteria like Golgi bodies, then other ribosomes and all, other things are there, but Golgi bodies not there, as well as these things also absent, endoplasmic reticulum, mitochondria. This specific organ we find in case of eukaryotic cell, but bacteria lacking this thing. But it contains mesosomes, ribosomes, chlorobium vesicles, gas vacuoles, and intracytoplasmic inclusion bodies. Besides other things, these are some main components, organelles present in the cytoplasm. Okay, now organelles and inclusion bodies. You have answered this question in your biology classes in your higher secondary program. In your school days even, no? Inclusion bodies and organelles. What are organelles? In a cellular organism, it has organelles, it has inclusion bodies. Organelles are?
Structural entities present inside the cell that control the function of the cell. That are engaged in various functions of the cell.
What about inclusion bodies?
They are not much.
Because cytoplasm having both these things in a cell, and in any cellular organism having organelles and inclusion bodies. Mitochondria also having important function, inclusion bodies also having important function. Functionally, both are important. Without this, cell cannot survive. It has certain function. Both inclusion bodies and organelles having some function. For functionally, they are present in this cytoplasm. But what way they differ? See, inclusion bodies and organelles, these are some structure that are present in the cytoplasm of the cell. It provides various functions. Both are essential. Inclusion bodies are non-living things, and organelles are living one. Now answer the question: mitochondria living or non-living? Endoplasmic reticulum living or non-living? What is the function of mitochondria? Powerhouse, ACB, Assam State Electricity Board. Power generation. What is the function of ribosome? Protein synthesis. Now there are certain organelles, there are certain structures which are living in nature, some are inert in nature, but they are very much essential for biological activities. Like gas vacuole, is it living one? Inclusion bodies, that is the metachromatic granules, these are non-living things, but it requires for certain activities. So that way, living one and that is the demarcation only, organelles and the inclusion bodies. The plasmid. So one important thing that I want to tell you. What the distinction between prokaryotes and eukaryotes? So far genomic structure concerned. What is gene?
Genes are functional packets.
Nucleotide sequence.
Nucleotide sequence, gene. What happened? Very ordinarily you can define gene.
Genes are the packets that contain the genetic information.
Genetic information, yes, very ordinary definition. Means what information we need. All information coded.
In the form of basic.
One gene code for one thing, one information generated there. And chemically what is gene? Gene is a nucleotide. Nucleotide sequence. What is there in a nucleotide?
Basis.
What are the things? Three things are there. One, sugar, nitrogen, and much better than me. Okay, it is having three basic components, and these three basic components constitute one nucleotide, and this is known as gene because it conserves certain information. It has certain information of protein synthesis. What type of protein? 20 amino acids. In this way, all information stored there. It's nucleus, or it's nucleotide, and this is known as gene. Then what is chromosome? Chromosomes are
Carries genes. Hereditary vehicle.
now you have defined gene, then you can define chromosome.